So hello everyone and welcome to Tamali's classes. In today's class, I'll be talking to you about a very interesting and important topic for CSIR-UNIT Life Sciences exam. So today's topic is RNA synthesis and processing. Do watch the video till the end because at the end, I will be giving you many questions that will be helpful for you to uh, conclude the topic. So watch the video till the end, and if you like the class, do click on the like icon, share it with your friends, and do not forget to subscribe to my channel for more such videos on life sciences, general biology, and on science topics. So without any more delay, let's start the class. So first of all, I'll talk to you about RNA synthesis and what it is. So in molecular biology, there is a term known as central dogma. This central dogma means how the DNA has been converted to RNA and RNA to protein. So we know that in human body, proteins are very, very important. So how these proteins are being formed is it's not being formed directly from DNA, but first DNA is uh, converted to mRNA, then mRNA to RNA, and then RNA to protein. See, mRNA, RNA, why I told you that because at the first it's a premature mRNA formed, and then it leads to the formation of proteins. So there are two steps out here: one is transcription and one is translation. So when DNA is converted to RNA, or you can say mRNA, it's called transcription, and when mRNA is converted to proteins, it's called translation. Today's class we'll be talking about the transcription process and also how RNA is being processed. That means how premature mRNA is forming, or you can say pre-mRNA is forming mRNA and the later stages. Okay, so in today's topic, we'll talk about transcription, RNA processing, and about how it is being transported. So now, what is the role of RNA in gene expression? The RNA is very important because we know, as I told you before, that proteins are very important for our body. So if a DNA is not converted to RNA in a proper way, and if the instructions, see, DNA converted to RNA means what? There are some instructions in the DNA which is being understood and taken forward to the mRNA, or you can say RNA. So this information has to be converted into a in a proper way, okay, into a proper molecule, or you can say protein, for it to be expressed in the body. Okay, therefore, if this process is not done properly, it will directly affect our life because proteins will not be formed in the correct way, and also the right proteins will not be formed. So it will cause problems, or you can say diseases in our body. Now let's see. So RNA will translate, and regulate the genetic information encoded in DNA. Because what does DNA contain? It contains all the genetic information required for our processing in our body. Okay. Now, first we can see in the diagram out here that double-stranded the DNA is there in the first one. So what's happening? Basically, I'm just telling you the basic steps out here. The basic steps is that DNA is first it is being unzipped. That means the DNA is double helix. We know that. So the first that the DNA helix is being unzipped. You can see out here. It will be unzipped. Okay, this double helix structure. Once it is unzipped. What will happen? RNA nucleotides will come and approach this unzipped DNA. Okay, so it will come and approach, and it will add the nucleotides. So once it adds the nucleotides, it will lead to formation of mRNA. Okay, transcripts of mRNA. So this transcripts now will move from the nucleus to the cytoplasm, and in the cytoplasm there are ribosomes. So once it moves to the cytoplasm, so there will be a pattern formed with involving your ribosome, then your mRNA, and also tRNA. So there tRNA, mRNA, rRNA, all will be there. The tRNA, it will bring some amino acids, and with the help of those amino acids and mRNA, the message in the mRNA, it will lead to formation of proteins. This step is it is translation, will not be discussed in today's class. Okay, so let's talk about the machinery involved in the transcription process in prokaryotes and eukaryotes. We'll do the process, all the process in details. Right now, I'm just giving you the intro. So in prokaryotes, first of all, in both pro and eukaryotes, you need a DNA template strand, which will be used for conversion to mRNA. Okay, and then the enzyme for prokaryotes is only RNA polymerase, but for eukaryotes there are three RNA polymerase 1, 2, and 3. Also in the prokaryotes there are no transcription factors are required, but in eukaryotes, since it's a complicated uh, process. So there are some transcription factors which are involved. And also now in the prokaryotes, there are some regions known as pre-mRNA box. Okay, this box has 6 nucleotide sequence, also known as TAT sequence, also known as pre-mRNA box. So this box is present 10 base pairs upstream from the site where transcription will actually initiate. Okay, but this type of pre-mRNA box is not present in eukaryotes. Eukaryotes has different promoter regions known as the Tata box and the TAT box. Okay, now this is the basic. Again, I've shown the diagram out here. In prokaryotes, basically I'll just say you, but in details we'll discuss in the future slides. See out here, first we have out here the DNA. Okay, so DNA is unzipping. Okay, so once it unzips, what will happen? RNA polymerase will come and bind to it, and then there will be a complex being formed, and this complex will lead to addition of new nucleotides. And once it's adding, what will happen? Slowly and slowly, the RNA transcript will be forming. So basically, let me tell you that the transcription, when the nucleotides are added in the direction of 3' to 5'. Okay, so RNA polymerase will move from 3' to 5' of the DNA template, DNA strand. Okay, the strand which it will be using to form a new transcript, mRNA transcript. But the transcription actually occurs from the transcription is forming from the direction is the 5' to 3'. Transcription is occurring from 5' to 3', but the nucleotides that are being added, the RNA polymerase is moving from the 3' to 5' of the DNA strand. Remember that. Now coming to the eukaryotic part, you can see the eukaryotic part out here. In eukaryotic part, this is the again chromosome out here. DNA is there, and there are two type of regions present. One is the introns and exons. Introns are the non-coding regions which do not code for any information, which do not contain any information, genetic information. But exons are the coding regions which contains the genetic information. So during transcription, remember, only in the regions which has the information will be coded. Only the exons will be coded, not coded, but only the exons will be taken into the process, but introns are not required for this process. So in due process, we will see that once the transcription is occurring, after the transcription occurs, is a process known as splicing. Splicing means to remove the introns, because we do not need the introns. Introns do not contain any information. So in the splicing process, the introns will be removed and only the exons will be taken, and it will be forming the complete mRNA from the precursor mRNA to a mature mRNA. Okay, so that is the process in eukaryotes. That's why it's a complicated process, because it involves the processing part also. So we will see the process in details. So let's talk about the prokaryotes first. So as I've told you before, in the prokaryotes, what happens? First of all, the DNA is unzipping. Okay, so the DNA, once it unzips, there are two double helical, right? Those two strands are there. So one strand will be used as a template strand, which will be used by the RNA polymerase. So remember, in prokaryotes, there's a bubble-like shape formed. Okay, so that is also known as a transcription bubble. Why bubble? Because when the strand will separate, the upper one is not required. The template strand will, when it separates, it will just see unzipping doesn't mean it will unzip full. So in the center where the sequence where the transcription will start, there it will unzip and it will separate and it will form a bubble-like shape like this. So and then the lower part, or you can say the strand from 5' to 3', it will be used for the transcription process. So you can see out here, this is the active site where it is unzipping, and the RNA polymerase, this will come and attach with in that site, and while it's keeping on adding from 5' to 3', the nucleotides, the DNA is converted into the mRNA. So the RNA transcript is forming from the 5' to 3'. Okay, so it will keep on moving. The RNA polymerase will be keeping on moving from the 5' to 3', and the new transcript being formed, the new nascent RNA being formed, it will be moving from the 5' to 3'. So the transcript is being formed in the 5' to 3'. It will be keeping on moving out of the bubble, but the RNA polymerase is moving from the 3' to 5'. So basically, you can see here, it's moving from the 3' to 5'. It will move like this, okay, and the RNA polymerase will be formed like this. We'll keep on, you see from here, first RNA polymerase comes here. So first is one thing is one mRNA is forming out here, one sequence, and it keeps on moving like this, and it keeps on adding like this. So moving, adding, moving, adding. So this way, slowly it will come out of that bubble and it will form the mRNA transcript. Okay, so this is the process that is occurring. Now coming down, you can see the RNA polymerase structure. See, this is the transcription site. That means from here, the transcription will start. That means from here, RNA polymerase will start adding the RNA, start adding the nucleotides. But this sequence is the promoter. That means this is the identification. So this is the TAAT region, okay, which is also called as the Ribnox box. As I said, it is -10. -10 means it is 10 base pairs upstream of the initiation site. So when initiation site, when it goes upstream of the initiation sites, it comes as minus, and when it goes downstream of the initiation site, it goes as plus. So you can see here, after this point, it is +1, but here as soon as it's going up, it's -10. So -10 region is the Ribnox box outer side, TATAT region, or you can say 6 nucleotide region out here. Okay, it contains 6 nucleotides out here, and above that there is another one in -35 region, TTGAACG. So these are all the upstream regions. So you can see the promoter regions, and there's also a factor known as rho factor, which is involved in the promoter region. Okay, remember this for the prokaryotes. Now, what are the polymerases involved in the eukaryotes and prokaryotes? So you can see in the eukaryotes part out in the top that RNA polymerase is 3. Okay, the pol 1, pol 2, and pol 3. So pol 1 out here, it will transcribe only rRNA, but not the 5S. Okay, and RNA polymerase 2 will be transcribing mRNA, S in RNA and miRNA, and RNA pol 3 will be transcribing tRNA, 5S RNA, and small RNAs. Location you can see RNA polymerase nucleolus, and the other two are in nucleoplasm. Now, if you see in the prokaryotes part, prokaryotes there are again 4 subunits: alpha, beta, beta dash, and rho. Okay, sigma. Sorry, sigma. So this is all about your subunits. Okay, you can say this is rho also, as it looks like rho. You can say rho also. So what are the functions? These are all, they are functions, all are against initiation and elongation. Okay, and also since rho factor is involved in recognition, so in prokaryotes you can see rho. Okay, you can say this is a rho, you can say just rho. So this subunit is known as rho. This rho will help in the promoter recognition, as I have told you in my previous slide, because it's acting as a uh, recognition factor. You can say factor recognizing the promoter region in prokaryotes. Okay, so these are the 4 subunits for prokaryotes, and the above ones are the RNA polymerase 1. So one thing I'd like to tell you, okay, when I will tell you about the differences between the transcription in prokaryotes and eukaryotes, I will tell you about the subunits, number of subunits involved. Okay, so otherwise I can tell you right now only. Wait. Okay, see here one thing. The RNA polymerase. Okay, you can see this point out here. Since I've spoken to you about RNA polymerase, I'll just mention to you this. In the prokaryotes, RNA polymerase has 5 subunits. Okay, and in the eukaryotes, RNA polymerase has got, since there are 3 types, so there are 10 to 15 subunits. So you can see RNA polymerase 1 has 14, RNA polymerase 2 has 10 to 12, and RNA polymerase 3 has 12 subunits. Okay, so this is just, I just wanted to tell you about it. Now coming back to the process, the next is formation of the initiation complex. So when initiation starts, okay, when it starts, now we talk about the eukaryotes part. It's a complicated process. So first of all, when there's a region out there, okay, see, there's a region out there, you can see here, but I'll show it to you. So you can see out here, this is the region, okay, where the transcription will start. The promoter region is there. See, this is the -30, you can see pre-mRNA box, okay. This region, this is the transcription start site. So in this region, first, the pre-mRNA box is being identified. Okay, as soon as it's being identified, the promoter region, okay, it's being identified, what comes and binds to it? TF2D and TBP. TBP is Tata binding protein. So first of all, the Tata box is identified by the Tata binding protein. It will come and bind to it, and the Tata binding protein is a subunit of TF2D. This is a transcription factor. TF2 is transcription factor. The factor is D, TF2D. So first of all, the data box is being identified by the TBP protein. TBP protein is a subunit of TF2D. The first TF2D and TBP together, they will come and bind to it. And after that, the other transcription factors like TF2B, then TF2F, TF2E, TF2 B and TF2H, they will all come together and they will form the initiation complex. So when TF2H comes, TF2H is the helicase. So when TF2H comes, it will start unzipping the DNA. Okay, this is the helicase. So now coming back, so this is what I told you. So all the recruitment happens, all the transcription factors comes and bind. It forms the pre-initiation complex. TF2H opens the DNA. Okay, and then the transcription starts. Now, as I've told you, in the eukaryotes, in the primary, when the DNA is being converted to mRNA, in the primary RNA transcript, there are both introns and exons being converted from the DNA to the mRNA. But we do not need the introns. We need only the exons because the exons contain the actual genetic information to be translated. Okay. Now, for that, what will happen? We got the mRNA, but we need to remove the introns. That's called splicing. So now we have to process the RNA in such a way that we get a proper mature RNA. Okay, so that will include the processing stages. So first we will do the splicing. So splicing will involve spliceosomes. Okay, so before that, let's talk about a bit about elongation part also. See, elongation, what's happening out here? As I told you before, RNA polymerase will come and bind. Okay, and it will be keeping on adding the nucleotides. Then what will happen? The transcription will occur, the elongation will occur, and the DNA, the nascent transcript will be keeping on forming. The direction of synthesis is 5' to 3'. Okay. Now, what is happening is termination. So for termination to occur, okay, for termination to occur, what is required? There are some sites required. So there are two things. Remember, two termination stages are there in for prokaryotes. We're talking about prokaryotes out here. Okay, now for prokaryotes, in termination, there are two factors, row independent and row dependent. So for row dependent, I'll just show you the diagram with row dependent out here. So the diagram out here. For row dependent, this row factor will come and first bind. Okay, you can see here. The row factor will first come and bind here. Sorry. Okay, it will bind here and it will keep on moving upwards. And once it comes and faces the sequence, or you can say the structure, where it can no more proceed, it will end there. Okay, so row, this is the row dependent. See out here, the row, it is going on carrying going up, and then when it reaches a certain point where it can go further, there the termination occurs. Now coming to row independent, that means where row factor is not there. So there is a sequence known as GU rich stem. So what will happen? The sequence will be keeping on forming and forming and forming, and then certainly again a sequence will come. There will be a sequence where GU rich stem will form a hairpin loop structure. Okay, so when this hairpin loop structure is being formed, or you can say when it reaches a sequence where no more it can proceed, where no more nucleotides can be added, what will happen? That sequence will pull itself and form a hairpin like structure. So once that sequence is reached, it will lead to termination. So this is the row independent termination. Okay, so I've mentioned, you can see I've highlighted the part CNG region, rich region nucleotides and a stable hairpin like structure. Okay, so this is about the termination. Now, as I told you, RNA processing. So after the RNA is formed, we need to process it. We need to make it a mature one. So first, I told you there has to be a step known as splicing, removal of introns. There are other steps also, like your capping, 5' capping. Okay, so capping means what? They are adding guanine nucleotides in the beginning of the RNA. Okay, so what will happen? It will seal the mRNA. It will seal it, and no more nothing else can come and add to the mRNA. It will not it will not, no, nothing else will add. The mRNA will stay intact. Okay, other step is your polyadenylation process. Outer what happens? Poly-A tail, that means many A's, around 100 to 200 A's are being added at the end of the RNA. So it's called addition of poly-A tail. So this is called as polyadenylation. Now since capping is occurring in the beginning, so 5 dash is adding, okay, 5 dash capping. And adenylation is occurring at the end. So it's in the 3 dash end, known as 3 dash polyadenylation, okay. These two is there for keeping the mRNA intact. Next is splicing, as I told you, removal of introns. Then we have got RNA editing, where there are some modifications occurring in the base, in some bases, and also at the end quality control and export. See in this diagram I told you about the spliced RNA. See out here. Also there is this one. You can see the diagram. See the poly A tail at the end part and the 5 dash cap in the beginning part. Okay. And this untranslated region is the introns, because it has not been, the information has, there is no information. So it is of no use. So it's called untranslated region. Okay. So only exon 1, you can see out here, from here only exon 1, exon 2 and exon 3. So only these three regions have been taken: exon 1, exon 2 and exon 1 blue, pink and the orange one. Exon 1, exon 2 and exon 3 has been taken, and the intron 1 and intron, you can see both the introns have not been taken in your splicing. Now we'll talk about the uh, lysosomes which are actually taking part in this. Okay, so what is lysosomes? Lysosomes is a sequence. It's a sequence. You can say it's a association between the snRNAs and proteins. Okay, so when snRNAs, okay, it's a small nuclear RNAs and proteins, they come together, they form snRNPs, or you can say SNRPs also, SNURPs. Okay, so they are basically, they are having U1, U2, U4, U5, U6 sequences. So what is happening? These regions, they are helping in the splicing part. Okay, they are adding to the U1 is adding to the intron. It is going and attaching to the intron. It is removing it. So the first one, second one, that way it's going on. Is functioning together. Okay. Next is GUAG rule. GUAG rule means, see, the introns, they have got the region in the beginning. There is a, in the 5 dash and the 3 dash end, there are some certain regions of GU and AG. See, when mRNA, when DNA is converting to mRNA, we know that DNA has ATCG, but RNA has U, okay, AUCG. Instead of T, it has got U. So so that's why when the mRNA is being formed, the introns, they have got certain region in the beginning of GU regions, and at the end it has got some region AG. Okay, so 5 dash and 3 dash. So this particular sequence is being identified by the spliceosomes, and it knows that this is the introns and this needs to be removed. So they will identify those regions and they will go and attach to it, the spliceosomes. Okay, once it goes and attaches to it, what it will do, it will cut the, splice the exon part. Okay, that means beginning of the intron, you will cut it. It will remove the exons and it will bind to the intron. This, from one intron to another intron, it will fold itself and form a lariat like structure. Okay, it's called lariat formation. I will show you in my future slide. So it will cut, bind to both the corners of the introns, and it will form a lariat like structure, and it will remove the folded introns. Okay, and it will form the proper mRNA, or you can say spliced mRNA, which will contain only the exons, not the introns. So there's another step known as alternative splicing. What they do is they do mix and match like process. What it is exactly, I will tell you, and it is important because it will increase the diversity of the proteins formed. Okay, let's see about it. Now, this structure, don't get afraid of it. I'll explain to you properly. So you see here, see out here, this is the mRNA being formed. Okay, this transcript. So this is the exon part. We need this one, we need this one, but we do not need the introns. So what will we do? Okay, just a minute. My laptop battery is going down. I'll just link my laptop battery charger. All right, so we are done now. So let's start. So if you look at the structure here, okay, so if you look at the structure here, you will see that here, see here, we do not need this introns part. Okay, so in introns part, see the structure out here, GU AG region, you can see here. So this is being identified and it is being spliced. Okay, 5 dash and 3 dash, and see the exons are being removed and it is forming a lariat like structure. This is the lariat structure. Why? How? this the end of the intron, the 5' end, it is going and attaching with the branch site. There's a branch site present. Okay, it will go and attach to the branch site, and again here there will be a splicing. Then this is a lariat-like structure being formed. So the exon 2 is again coming together with the exon 1. Okay, so this is there. Now we're talking about alternative splicing. Look at the right side diagram now. So as I've told you, it is being this is being cleaved. So we have the 3' cleavage out here and the polyadenylation, so that no more other nucleotides, or you can say other anything comes and attaches with the mRNA transcript. We need it proper. We need it in a proper way. So now once this poly A tail is coming and attaching and the splicing is occurring, now there's another term, alternative RNA splicing. That means what are the other ways of splicing the RNA? Here, first of all, there's two possibilities. One is either this exon 1, exon 2, and exon 3 and poly A tail, they come together and they form a proper mRNA, okay? Or other way it can be that maybe the exon 1 and exon 2 can come and the poly A tail can come. That means exon 3 is not coming, okay? But both are okay, but you can see in the first one we have all the coding regions, exon 1, exon 2, exon 3. But in the other one we have only exon 1, exon 3, and we do not have the exon 2. The number 2 one, exon 2 has been removed, or sometimes exon 3 might be removed, or might be exon 1 might be removed. So there are many types of splicing. So when many different forms of mRNA can be formed, it's called as alternative splicing. Okay. Now, RNA editing. So as I've told you, sometimes this substitution might occur. That means sometimes a you can see there might be some insertion or deletion. Like okay, out here you can see the first one. In first case, C is being replaced by the U. Okay, you can see when CAA was, it was glutamine, but when C was removed and U came in, it became a stop codon. UAA is a stop codon. Okay, it means to stop. And again you can see out here A to I. Sometimes A can also be replaced to I. So when CAG was, it was glutamine, but when CIG occurred, it became RG9. So what's happening? Different type of proteins can form because of RNA editing, and the enzymes involved after is ApoBEC1, and here this is ADAR2. Okay, so ADAR is adenosine deaminase. So these enzymes are also involved in this RNA editing process. Now this can lead to protein diversity. Okay, it can help in gene regulation or cellular function, but sometimes it can also lead to formation of some diseases. Okay, it might lead to some diseases which are not welcomed, okay, in the body. So there are some good and bad things of RNA editing. Okay, now you can see out here the lower part, you can see the proteins part out here. The blue ones are unedited ones and the red ones are edited ones. So this part, sometimes edited ones might not be good for your body, so it can lead to some disease development. Okay, now polyadenylation, as I've already told you. So what is happening out here? There are some A's are being coming and adding. Okay, the poly A tail is coming and adding at the 3' end of the mRNA. Okay, so poly A tail is being done by addition is done by the poly A polymerase. So what's happening? It is important so that the RNA is intact, it is stable, no more anything is coming and attaching. And also when poly A tail comes and attaches to it, it forms a proper pre-RNA to mRNA is processed, and now this is ready for being transported to the cytoplasm, so that for what? For translation to occur. Okay, as we know that transcription is occurring in the nucleus. Okay, in this nucleus, and then it is moving on to the cytoplasm. Okay, and then for translation to occur, it is moving on to the ribosome and cytoplasm, and then the translation is occurring. So for this nuclear export, this polyadenylation is important, and also for the maturation and functioning also. Now types of RNA. So we know that many types: mRNA, tRNA, rRNA, snRNA, miRNA, and siRNA, and incRNA. So what are the functions? mRNA, we know that mRNA contains the information for coding, for translation, that means for proteins. It contains the information that to be translated to form proteins. tRNA will bring in the amino acids which are being read. Okay, and then rRNA, it contains ribosomal units which holds the mRNA and welcomes the tRNA. snRNA is involved in RNA splicing, as I've told you, in spliceosomes. Then we have miRNA and siRNA. They are again utilized in gene silencing. We will do that in later classes. and INC RNA is regulation of gene expression. So I've told you about the transport again. See, first when mRNA is being formed, it is being transported from nucleus to cytoplasm, because we know in the nucleus there are some pores present in the outer part. So to the nuclear pore complex, from the nucleus to cytoplasm, the mRNA is being exported. I can say transported. So there are some export factors, NXF1, which is being involved, and also the snRNAs and tRNAs are involved in the processing part, as I've told you, in splicing and recycling, and also at the end quality control. I was keeping on saying so that nothing comes and adds more to the mRNA. That means the RNA should be pure. It should not be degraded, or there should not be any defect in the RNA being formed. So for that, quality control is being done. If there is any defective or degraded RNA present, it will retain it. It will stop it from being transported for the translation process. Okay. Now, the differences between prokaryotes and eukaryotes. So as I've told you, in the prokaryotes, it occurs in the cytoplasm. Remember, transcription occurs in the cytoplasm in prokaryotes. But in eukaryotes, first it occurs in the nucleus, and then from nucleus it moves on to the uh, cytoplasm and ribosome. And then in prokaryotes, it is not so complicated. Why? Because the DNA is not attached to any histone proteins. But in eukaryotes, DNA is attached to histone proteins. So for initiation to occur, it's a very complicated process. It has to first get removed from histone, and then the initiation complex will come and bind. It's so complicated. So this one, for RNA polymerase, it will synthesize one RNA polymerase will synthesize all types. But in eukaryotes, there are three RNA polymerase: RNA pol1, pol2, and pol3, and they will synthesize the rRNA, mRNA, and tRNA respectively, as I've shown you in the previous slides. So in prokaryotes, it is polycystonic. That means only one mRNA will code for multiple proteins, or you can say one or more genes. But in eukaryotes, since it's complicated, so out here only one gene is there. It's known as monocystonic. That means one mRNA will code for only one protein. Okay. Then for prokaryotes, RNA polymerase has 5 subunits, and for eukaryotes, RNA polymerase has 10 to 15 subunits. RNA pol1 has 14, RNA pol2 has 10 to 12, RNA pol3 has 12. Okay. Now, transcription factor, as I've told you before, in prokaryotes it's not required, but for eukaryotes many transcription factors are there: TF2D, TF2E, F, H. Okay. Then B. Then for prokaryotes, promoter site is always in the upstream. Even in eukaryotes also it's in the upstream, but certain times in, like for polymerase 3, it is an exception. Okay, it can be downstream also. Then for the prokaryotes, promoter region, it has got Pibnox box, but Pibnox box is not present in the eukaryotes part. Okay, it is, and the eukaryote it is -35 upstream, and for promoter it is -10 upstream. Okay, right? Now, why it is RNA processing is important? I've already told you before. Once more, that RNA synthesis is very, very important because it plays a role in gene expression for the formation of proteins. If proteins are not formed, then gene will not be expressed, and you know that that is not at all a good thing for our body, because proteins are important, and proteins are regulating our body, entire mechanism and everything. So for monitoring, functioning, regulation, everything. So RNA processing is very, very important. So now there are some questions I've given out here. Around 8 questions are there for you to solve. I can solve it, but I will not solve it. I want you to solve it. So there are some questions out here which are new, I know. Like question number 2. See, from here some questions have already come for your CSIR previously, and some questions I've added out here to clear your entire concept on this topic. So the second question out here, I'll just say this because it's not there. I've not told you about it in the class. A dash helicase is required for stripping the spliced mRNA from the spliceosome. So after the mRNA is formed, the spliced mRNA has to be stripped, right? It has to come out. The introns have to be removed. It has to come out. So there's a helicase which is required out here for stripping the spliced mRNA. So what is that helicase? It is PRP22. Okay, I'm just giving this answer, PRP22. Now the other questions out here, the statements about the 5' cap, role of 5' cap, RNA splicing, lariat structures, okay, type of intron, everything I've told you. Okay, the type of introns, again there are many types of introns are there. So which type of introns are involved in formation of lariat structure, and the name of type of intron which needs the lysosomes to be spliced. So this all you have to do and give your answer. Comment your answer in the comment box, and I will tell you whether it's a right answer or a wrong answer. And also, if you have any more questions, do let me know. I'll try to clear these questions. And if you have any doubt, please let me know. So this ends our class today. And if you have liked the video, do click on the like icon, share it with your friends, and do not forget to subscribe to my channel for more such videos, because in my upcoming videos I'll be coming up with more very interesting and important topics of CSIR, and also I'll be discussing some important questions with you. So see you again in my next video. Thank you so much for today's class. Bye bye.